We should start. Are all the speakers here, by the way? We have two, I know. Okay, well, hopefully they'll come in. Okay, you can see the format up here. Welcome to the Genome Editing section. It's going to cover a wide variety of things. I'm going to start by giving a very top-level view of what genome editing is, the various techniques. And some of the limitations perhaps of using genome editing directly as a therapy, and then focusing then on what is more immediately applicable in using genome editing to find new targets for conventional therapies. And I will talk at the very end about a technique that we're developing called protein interference, which has similarities to other genome-wide screening techniques. So the next speaker after me will then talk about ways of improving delivery, which is a major limitation at the moment. And then after that, you get more detail around what the CRISPR system is. And after that, a talk around synthetic lethality using genome-wide synthetic lethal screens using CRISPR. Okay. So, target ID and validation. So, conventional drug discovery is very much centered around specific targets these days. Now, defining what a good target is, and then actually drugging the best targets is a major bottleneck for therapeutics. And editing genes directly with genome editing, is very exciting. Everything is druggable if you can edit it, but delivery there is a major problem. So I will touch on all these areas. So I've been interested in this continuum for probably about 20 years now, and that's converting information on human genetic variations into new targeted therapies, whether you want to call those personalized medicines or precision medicines. There's a lot of arguments around what the best thing to call that is. If you're a GP, they think they've been doing personalized medicine forever, and they're right. So maybe a more accurate description is precision medicine. So that's getting the right drug to the right patient at the right time. Revolved around a good understanding of what good targets are, and some of the best targets will be those that are mutated or variant in the population. And I've been very interested in using genome editing techniques to sort of translate that information into new therapies, a very conventional path rather than direct modification of genomes. And that's driven largely in part by, at least the early techniques, we would never have envisaged there being efficient enough to do that in vivo. So what I focused on really was using the techniques in vitro to create in vitro disease models that reflect patient genetics, creating essentially patients in a test tube that you can test your targeted therapies against, see if they work against those presumed patients that you think might benefit the most. But also there's a role for genome editing knockouts to understand what the genome is. We don't know still what most of the genes in the genome do. So there's a real need for a functional human genome project too, and that's something that's really being kick-started by CRISPR. Now, of course, the excitement of CRISPR and the gains in efficiency that has is maybe we can start to think about modifying human genomes directly. Now, the various techniques have evolved over time. I started genome editing many years ago now, probably in the 90s, using techniques that are based around homologous recombination. And that's on the very left side here. Now, these techniques work brilliantly in mice, making transgenic mice, often using just plasmids. This is because you can engineer homologous recombination is actually quite efficient in embryonic stem cells where you start to create transgenic mice. But if you want to engineer somatic cells, these are cells growing in the laboratory, homologous recombination as a DNA repair mechanism that you're piggybacking on essentially is almost shut off in somatic cells. So the big leap to do somatic genome editing came about with RAAV, which is a virus, a single-stranded DNA virus. And there's something about this virus being single-stranded that basically turns back on homologous recombination. Now, I say turn it back on, it's not brilliantly effective either, but it's a lot better than plasmids. And so this was fine to engineer cell lines, but no one ever thought to use this really for correction of diseases directly in vivo or even ex vivo. But it's a very simple technique. It's just a piece of DNA with homology to your locus with a change that you want to engineer in. And at a certain frequency, a pretty low frequency, it would basically flip into that locus. And if you want to knock out genes, you have to do it again. So to make a cell line, it would often take at least 6 months, often up to 2 years, even using this technique. So a real jump in efficiency came with nucleases. And the first ones were zinc fingers, and then following that, talons. And basically, these create a DNA break at a specific locus. The specificity is dictated by the zinc fingers in the talon sequence, and then cut by a nuclease. And these were useful when you create DNA breaks. That has to be repaired in some fashion very quickly, otherwise the cells die. This is great for knocking out genes. So basically, when you cut the DNA, the ends get chewed back a little bit and then stitched back together using a different DNA repair mechanism called non-homologous recombination. And if you give another piece of DNA in trans that has homology, you can get knock-ins, but the rate of knock-ins is much less than knockouts. So the efficiency of non-homologous end joining is much higher than recombination. Now, CRISPR is another step forward above that. Similar idea of creating targeted DNA breaks, but much simpler to engineer. So you're not changing the sequence of proteins now to get specificity to your locus. The specificity is dictated by a piece of RNA, a nucleic acid itself, so very easy to engineer to any particular locus of interest. Now, the same issues around knock-ins and knockouts are there for CRISPR, roughly 100-fold less for knock-ins. And so that, if you think about what you want to do in therapy, that has a bearing. So if the most efficient technique is knockouts, how relevant is that to correcting diseases? Well, most genetic diseases are already loss of function, so what we really need to do is correct them. So that's not going to be the most immediately applicable use, is knocking out genes, but there are a few examples, and one was given by Carl June yesterday around knocking out the HIV receptor so you can get resistance to HIV. But by far the larger group of things we want to do is correcting genes. So efficiency of delivery is going to be absolutely key. And many diseases like cancer, especially where you have to, if you want to correct cells, you're going to have to correct the majority, if not all the cells. It's not really going to be feasible. So it's really only applicable to diseases where a small percentage of transduction will basically ameliorate the disease. So it's things like hemophilia, for example. And it'll be aided dramatically by privileged sites, so sites where you can give very high local concentrations, such as the eye of the CRISPR vectors, or the liver, where you can give systemic administration, but basically things get concentrated in the liver because that is basically the body's garbage disposal site. So, how many diseases are going to be conforming to that? So it's pretty low. And so most people are focusing on ex vivo modification. This is eminently feasible, but now the rate limiting step there is, can you engraft those cells back? So this is very much around stem cells. You would have heard several talks yesterday around giving back CAR T cells, where you essentially, giving back the cells is really not a problem, but many other tissue types may be a problem. Now, here, when you're doing ex vivo, the speed is not necessarily essential now, it's desirable. I would say the precision is probably more paramount, especially if you're looking at, as Carl June was describing yesterday, sort of universal donor CAR T cells. Well, you really, if they're going to be used in lots of people, you really want to make sure the genome editing techniques are really clean. And so maybe there's still a role for things like AAV in that setting, but I think 99% of the time people are going to use CRISPR moving forward. So if there are limitations on directly modifying the genome, this is certainly going to mean we're going to need conventional therapies still in the future. That's not going to change. Now, we have our own problems, though, when it comes to developing conventional therapies, whether it be small molecules or biologics. And this is very much around druggability. So the number of genes that are easy to drug, things like kinases, for example, which have deep pockets to them and they bind small molecules already that we basically just copy to develop more effective inhibitors. There's a very limited pool of these that are easy to drug. They're actually the best targets. Now, anything can be targeted with an antibody, but here the limitation is antibodies, macromolecules, do not get through cell membranes very well. And that may be fixed, but that's still a major limitation. And if you think about targets inside the cell, these are very much predominated by protein-protein interactions. So while there's only roughly 100,000 proteins in the genome, there are literally millions of different protein-protein interactions, each one of which can be a valid drug target in its own right, perhaps with its own unique mode of action. And that's really an untouched area. And if you look from a therapeutic perspective in oncology in particular, the number of genes we know are good targets. So if it's mutated to a high frequency, and many cancer genes are, you pretty much know that's a great target to go after. But most of those great targets are not directly druggable. Even if they're oncogenes, sometimes like KRAS, which is a gain of function in the pathway, but actually a loss of function in the enzyme, it's very hard to reconstitute activity using small molecules. Other targets are protein-protein interactions like c-Myc, and other things, a vast majority even, are tumor suppressors, and these are often deleted from the genome entirely, so they're not even in the cell. So how do you target something that's not there? Now, for some tumor types, all the genes that are currently understood to cause them are actually undruggable. So there's a major need to start to open up new druggable space. And so what do you do if your target is undruggable? There's basically two ways. There's one way that's very common right now, driven by genome-wide screening efforts using either RNAi or CRISPR. And that is basically going around the problem. So if your target is undruggable, is there something downstream that is conventionally druggable? But then you start to look at the complexity of signaling pathways, for example. It's very hard to determine what's downstream. Pathways are not pathways, they're networks. And so it's very hard to guess, and people don't guess anymore because the techniques are there to just go one by one and knock out every gene in the genome using CRISPR or suppress the message using RNAi. And the technique we're developing called ProteinI can do the same thing, genome-wide, but working at a protein level. And by working at a protein level, there's this other way potentially of drugging undruggable targets. I think there's actually more druggable space than we appreciate. They're just not as obvious. The druggable sites won't be as obvious as something like a kinase. They're probably very small, often probably fairly transient in the cell. So proteins are living and breathing entities in live cells. Pockets come and go all the time. And when you get a crystal structure, you just get one snapshot. And very often you can miss these small transient sites in crystallography. And you may not even think these are druggable a priori without knowing that it's doing something useful. So the key is to screen live cells for these potentially cryptic druggable sites. And this is what ProteinI is. And it contrasts with genome editing and RNAi quite clearly by working at the protein level. It's very useful to do genome editing and RNAi screens. But the limitation there is it can only tell you what a good target is. It doesn't tell you how to drug that target. So I'm very keen to take that next step forward to find a technique that can systematically not just identify the best targets, but also how to drug them. And we do this basically with small protein fragments. So we express on a single cell by single cell basis a fragment of a larger protein across the whole proteome with basically millions of different shapes, a different shape in a different cell. And that will elicit a response, a phenotype that you think is useful. And then you isolate the target to that peptide that you express that is creating that phenotype. Once you know the target and the peptide that binds to it, the shape of that, you have the beginnings of a conventional drug discovery program. We're not using these peptides as therapies. They will have the same issues as antibodies, but the shape, understanding the shape, will help us design small molecules with the same shape. Now, screening ProteinI, where we have millions of different shapes, it's a massively taller order to screen that number of things in phenotypic assays compared to CRISPR and RNAi. So we are starting to move towards these very high throughput screens. Initial low throughput screens on a range of pathways gave us very encouraging hit rates, but that's not really where we want to be. We need to be doing high throughput stuff. So following the lead from shRNA screens and more recently CRISPR genome-wide screens, where somehow you get the needles out of the haystack and determine, in a pooled fashion rather than doing well by well screens. You screen everything in one well and find the cells undergoing the phenotype. Now that can be done in several ways. It can be the biological selection, where the rare survivors are the needles, and you just look at the needles once the haystack's gone. Slightly more difficult is just physically separating the needles from the haystack. Or more recently, you can just use NGS, where you just leave everything where it is, sequence what's before and what was there before and what's afterwards, and perhaps you can find synthetic lethal stuff that way. And I don't have time to go through all the data on these various approaches, but here's just one interesting one on a very undruggable target called KRAS. Here we put 5 million different proteinized sequences into a parent cell line in which we can induce KRAS. Inherently toxic stuff is deleted out, then we induce KRAS and look for dropouts. And we're seeing very clearly dropouts in this system. And we need to go find the targets of these, but that's in process. So the last slide is just summarizing these various techniques. RNAi, CRISPR, and small molecule protein I. So all these techniques are good for target inhibition. Whether you want partial or complete is a bit of an argument. CRISPR, I think you'll find things that RNAi misses. Small molecules and protein I can do the same. But really the difference here is CRISPR can do activation as well as inhibition. And you can do a whole lot more things now with various variants of Cas9, which I'm sure one of the other speakers are going to. But the key thing is these knockouts do not find new druggable space. They don't actually even phenocopy small molecules. So removing a whole protein is not what a small molecule does. So that can often lead you down the wrong path. Now, whether we can actually find new druggable space is still ongoing. We have at least the theoretical ability to do so. We're getting good hit rates, but we have to now go find those targets and do they look druggable. Okay, so I'll end there and take any questions. This one over there.
Thank you. That was a rather complex talk in the sense, or quite different than I had expected. So just to make sure that I got your point.
Yeah.
So you're not using it for any in vivo or ex vivo application, the technique. You're using it, I'm reading Swiss Army knife, as a research tool to actually have a better way to identify targets and then later come on to small molecules with better druggability and better specificity.
That's right.
So it's as a drug discovery tool, not as a therapeutic tool.
Exactly, yeah. So my particular technique is protein I. Another talker here will talk about using CRISPR to find conventionally druggable targets. So this session is about genome editing as a direct therapy. I'll try to address some of the issues that I think it might have, but I'm not expert enough. I'm not working in that area to sort of address how well that field might go.
That's fine. Thank you.
Sorry, thanks.
Hi, excellent talk. I really enjoyed that. I just wanted to know how you think that protein I kind of sits alongside things like peptide aptamers and those kind of techniques, because I guess there's some comparisons which could be drawn. Yeah, because of the time limits, I couldn't really elaborate too much what protein I is. So that's a really good question. So peptide fragment or peptide sequence screening has been done before in some forms, very commonly like phage display, yeast 2 hybrid. They're very much around finding affinity reagents. And you can get affinity from very simple, well, actually no structure to it. You can get it from just simple primary sequence. And random peptides may have some structure, secondary structure possibly, but they won't have tertiary structure. And so what we need to do to find druggable space is describe three-dimensional space in a target. And I don't think you're going to get those commonly
from random peptides. So there are those libraries out there. People have even tried to screen random peptide libraries in phenotypic screens. The hit rates are really low. So you can get very good hit rates for binders, but not great hit rates for phenotypic hits. And so the key, I think, will be screening coding sequences, which nature has biased to have structures. Proteins are made up of subdomains and they're self-folding. So basically it's a subdomain fragment library across the whole genome. Now you can use any species in theory to do this. We're using human genome fragments, but one of the main ones we're using is a bacterial fragment library, and they come from extremophile bacteria, so they really have to fold their subdomains very rapidly to survive in deep sea thermal vents. So I think that, as far as nature can, has biased us to three-dimensional space in that setting. This will be the last question, I think. We'll have to move on after this.
Thank you. Nice talk. I was wondering whether you can use Protein I also to sort of, instead of doing evolutionary studies where you would try to improve on protein functions or make them more specific or whatever, to use Protein I to see whether you can create compounds that give new or better functions to already existing proteins.
Yes, that's a very, it would take a long time to answer all those fragments. So yes, I think there's going to be ways of drugging proteins that we don't predict. And I'm very keen when it comes to protein-protein interactions, it's not just to find peptides that break apart directly the interface. I think those would not yield small molecules at the end of the day. So I'm very keen to find sort of allosteric distal sites that would be often transient, hard to describe, that when you dock to those, and they don't have to be very high affinity to those sites, is change the overall structure of the protein, and then that breaks apart protein-protein interactions. So yes, I think we could find very different ways of understanding protein function, changing protein function, and perhaps we can evolve the sequences, the primary sequences we find with Protein I with further evolution to find better binders, and then that describes a better shape for a small molecule. But the size of the libraries we're screening is so massive already, millions, even billions, 10 to the 9. I'm hoping we've got all those possible shape variants in one pot already. But there could be some evolutionary aspect to changing those hits to be even better binders.